Hello everybody, it is week 9 of the board review class and we're continuing biochemistry and we're going to talk about gluconeogenesis, the savior of starvation. So again, you guys have all had this, so you should be fine with it. It's made to jog your memory, but I am going into this stuff, not like I did in endocrinology where I just kind of skimmed through it. We're going to go into this one a little bit more thoroughly because it's so important, right? This is type 1 diabetes. Gluconeogenesis is driven all the time. It's on all the time, so you need to understand this. Oh, we got to do our birds, so I don't think I showed you that one. That is a short-eared owl from last Tuesday. Very rare. Very lucky to get that. And that is also from Tuesday. That is a golden eagle, which is, it's just a little bit bigger than a bald eagle. A mighty, mighty hunter, much more vicious than a bald eagle. All right. A little where have we been? So we talked about glycogenesis or glycogen synthesis last time. And I'm just basically going to use this, and just you could read all the details here. But basically, glycogenesis is the building of glycogen that occurs during the fed state, where you can take extra glucose and make a storage form of glucose called glycogen. And the key to drive this one, or to make glycogen, is, and the rate limiting step is glycogen synthase. And glycogen synthase is stimulated by insulin, kind of indirectly. Insulin is it's not a steroid hormone, so it can't go through the cell membrane. So it can only kind of anchor itself to the outside of a cell membrane. And through a second messenger system, PI3K AKT system, AKT is the main effector, that stimulates this protein phosphatase 1, and that removes a phosphate from glycogen synthase, which activates it. So that's kind of the key point there. Now, after 4 hours is gone, your dietary glucose is gone. So we know that tissues like your brain, your red blood cells, they need glucose all the time. So after 4 hours, where do we get blood glucose from? If you're maybe like overnight fast, you're sleeping, you're not eating, where does it come from? Well, it comes from that stored glycogen that we just made in glycogen synthesis step. Now we just reverse that, but there is a different enzyme. And when glucose levels fall after 4 hours, insulin is no longer around to do anything. And so now we have our friend glucagon around at times when insulin is dropped. And glucagon will stimulate not glycogen synthase, it actually inhibits it, but it stimulates glycogen phosphorylase into action, and that is going to start picking those glucose monomers off that gigantic glycogen molecule and converting it, making glucose 1-phosphate, which goes to glucose 6-phosphate, which goes through glucose 6-phosphatase, it goes into glucose and it's released. So glucose binds to the cell or glycogen binds to the cell membrane and triggers a different second messenger system, the cyclic AMP pKa system. pKa will go stimulate phosphorylase kinase. Phosphorylase kinase will phosphorylate, not remove, phosphorylate glycogen phosphorylase, and that activates it, and then it does its thing. It starts breaking down glycogen. All right, so we have a whole video on that. Don't need to spend any more time talking about that. So in simplest terms, insulin activates glycogen synthase, which makes glycogen, while glucagon activates glycogen phosphorylase, which breaks down glycogen into glucose monomers for release into the bloodstream. All right, so what happens when glycogen runs out? Normally glycogen runs out after about 24 hours. Now what's going to happen? Well, now this is where gluconeogenesis comes in. It takes the baton from glycogenolysis, the breakdown of glycogen, and it usually kicks on at 24 hours. Why do we have to? We already know this. Red blood cells, 100% dependent. They have to have blood glucose. They can only run anaerobic metabolism. Renal medulla, 100% dependent on blood glucose. The brain is arguably 40% dependent on glucose. It can use ketone bodies in starvation, but it still needs at least some glucose. The lens and cornea of the eye, 100% dependent. Wouldn't that be nice? You can see the Gillard question coming from that, can't you? All right, so that's the star of this lecture, is gluconeogenesis. And this is daunting. This took me forever to make this. This is all checked with board biochemistry and physiology books, so everything is good. And what I'm going to do is just blow up little sections of this as we go through. Great thing to study before boards because it has all the key regulatory enzymes in there. But this is where we're going. So what is it? What's the definition of gluconeogenesis? Well, it's a metabolic process. It's run in the liver and the kidneys, specifically the proximal convolutubules can run this. The intestines can run it a little bit, but usually they don't talk much about that. It's the liver and is the main one by far. So hepatocytes. We said it takes off after glycogenolysis kind of drops the baton, runs out of glycogen for fuel. It takes over making glucose from scratch, and glucose is made from non-carbohydrate precursors. We're going to look at those in a second. And yeah, said everything there. So let's look at the three things that fuel gluconeogenesis first. And where does this, where do you even start this thing? And what is this thing? This is a hepatocyte in the liver. And this white is the interstitium, and there's a capillary right there. And the best place to start the discussion is right here, our friend pyruvate. And we know pyruvate, that should sound familiar because that's the end product of aerobic glycolysis. In fact, it's anaerobic glycolysis is the end product as well. So all glycolysis, the end product is pyruvate. And that's where we're going to start the story. And we just can't run back forward through glycogenesis. We have to run a different direction, or glycolysis. We're not talking about glycolysis, but everybody knows glycolysis, right? You had that in undergrad. So you can't just run backwards through glycolysis. We have to get, well, we can in a way, but we can't run pyruvate backwards into phosphoenolpyruvate. We have to do a side route to get back to phosphoenolpyruvate. So let's start talking about it, and we'll see how this goes. So the first thing to do is talk about the fuels. So there's pyruvate. There's three fuels we said, right? There's glycerol from fatty acid breakdown. That does not convert into pyruvate. That converts into dihydroxyacetone phosphate, actually further toward the making of glucose, further up the chain. So, but we're going to talk all the way about the very end of the chain and go all the way through gluconeogenesis. And so lactate comes from the breakdown, it comes from pyruvate being converted into lactate. Remember, red blood cells and white blood cells and platelets, they can't run anaerobic, or they can't run aerobically. They don't have a mitochondria. So they can only run glycolysis. And therefore, they run anaerobic glycolysis, and the end product, that pyruvate, is converted into lactate. And so that source, in fact, is the main source that feeds gluconeogenesis up until about 1 week. So lactate from red blood vessels, skeletal muscles, even if you're sitting in your chair, maybe 5% of skeletal muscles, the ones very deep inside, they're actually running anaerobic glycolysis as well. And so they release lactate. Lactate is always released if you can't go into the Krebs cycle. So that's where it comes from. There's an enzyme, lactase dehydrogenase, which converts it into pyruvate, and there's our start right there. So what else do we need to know about lactate? It enters gluconeogenesis as pyruvate. It comes from skeletal muscle and red blood cell anaerobic metabolism. It's the number 1 fuel source for the first week. It becomes the number 2 fuel source thereafter. So until you either get food for your fast or you die from your fast, it'll always be the number 2 fuel for gluconeogenesis. And lactate dehydrogenase is an enzyme. And notice these arrows on this are painstakingly accurate. I checked every single one of these with board biochemistry books. Leininger is the king, the gold standard of biochemistry. I also have Lippincott, so yeah. Great, so that's it for lactate. What about amino acids? Where do they come from? So again, about 5%, and that number varies, but a small percentage of skeletal muscle cells are always running anaerobic glycolysis, and they are also contributing to lactate here, we said. But I just wanted to throw that in. Amino acids, where do they come from? Well, they come from skeletal muscle proteolysis. So when you're fasting, you're going to break down skeletal muscle. And skeletal muscles are made of all kinds of amino acids, but they all go through a process, a transamination. They all have aminotransferases, which morphs them into one common transferable amino acid, and that's alanine. So alanine is released from skeletal muscle breakdown. And remember, we're into a fast. We're a full day of fasting now, so you are going to break down some skeletal muscle. And alanine is converted into pyruvate via alanine transferase, or ALT. Very common lab finding, right? They always check ALT to see. That's a good indication of liver function. Okay, there are other glycogenic amino acids. Glycogenic means they can go through gluconeogenesis, like serine, glycine, cysteine, threonine. They can also morph into pyruvate as well, and we won't worry about the enzymes that get them there. So pyruvate is a pretty popular starting molecule for gluconeogenesis. Side note, the TCA cycle amino acids, I think I have them on the next page. Yeah, here they are. So, and this is not a complete list, but there are some other amino acids that get loose from skeletal muscle catabolism and breakdown and float in the bloodstream, and they end up in the hepatocyte, and they can get in this broken TCA cycle. It's broken because it's not really a cycle, because acetyl A isn't driving this cycle. The cycle really starts during starvation at citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate, which is the end of the molecule. But it's interesting that glutamate or isoleucine or phenylalanine, they can enter this broken TCA cycle, be converted to oxaloacetate, and then something special happens. Oxaloacetate that's converted from the carbon skeleton of these amino acids, it doesn't have to go through this malate shuttle that we're going to talk about. There is a mitochondrial form of Pepsi K. I like that, Pepsi K. That is phosphoenol. That's what is that? Phosphoenolpyruvate carboxykinase. Pepsi K is better. But Pepsi K, a mitochondrial form, converts it into PEP right inside the mitochondria, and PEP is able to go in and out of the cell membrane like nothing, the mitochondrial membrane like nothing. And so it can just go on its journey through reverse glycolysis here. So that's all I'll say about that one. So mitochondrial Pepsi K converts oxaloacetate to phosphoenolpyruvate, which can leave the mitochondria without a transport. It just floats through that membrane like a ghost. So, pretty cool. Fun fact, skeletal muscle is the target of fasting-related proteolysis throughout the majority of fasting. However, once the fat supplies are gone, because glycerol is going to take the lead as fasting goes on. But once your fat supply runs out, the proteolysis comes back with a vengeance, and not only does it hit your skeletal muscles, it hits your organs, and that's what ultimately kills you during starvation. All right, what else? Glycerol is a little different animal, as we said. So glycerol does not go into pyruvate. You definitely should know a softball question like that. Where does glycerol enter the gluconeogenesis cycle? Certainly not pyruvate. So let's talk about glycerol just for a second. So during fasting, you have low glucose. There's a nucleus in the hypothalamus, the arcuate nucleus. There's neurons in there. There's a story in there. I'm not going to even bother you with it. Let's just say the arcuate nucleus senses decreased blood glucose, and that will stimulate the lateral horns of the spinal cord to release a barrage of sympathetic outflow. And sympathetics will hit the chromaffin cells of the adrenal medulla, and that we know what they release, right? That's epinephrine, norepinephrine, a little bit of dopamine, the catecholamines. And so epinephrine is loose here. So epinephrine binds to a fat cell, and through a classic cyclic AMP pKa second messenger system, the pKa stimulates the breakdown of fat, specifically epinephrine, and glucagon helps a little bit, but not a whole bunch. Epinephrine is the key. It stimulates hormone-sensitive lipase to release or to break down fat cells and release the components of a fat molecule, which is glycerol and 3 fatty acids. That's a softball question. You should know hormone-sensitive lipase will break down a fat or adipocyte into what? Glycerol and 3 fatty acids. Okay, it's stimulated not by glucagon but by epinephrine. So anyway, that's where glycerol comes from. Glycerol morphs. And when I say morphs, I mean is converted into, I like to just say morphs. It morphs into glycerol 3-phosphate. There's an enzyme, glycerol kinase. Glycerol 3-phosphate morphs into our friend, which is a intermediate or a domino in the glycolysis pathway and in the gluconeogenesis pathway. Our friend dihydroxyacetone phosphate. Great. Reversible enzyme, non-reversible enzyme, glycerol kinases. Great. And then we'll talk about this further when we get to it. So that's the story with glycerol. So just the facts on the slide. Glycerol enters gluconeogenesis as dihydroxyacetone phosphate. It comes from the process of lipolysis, so the breakdown of adipocytes, fat cells. It becomes the main fuel of glycolysis. After about 1 week, it takes over, and it will stay the main fuel. So in other words, glycerol will become the main fuel to keep you alive. And that kicks on after a week of starvation, and that will stay until you run out of fat. That's why those survivor shows, the people who have more fat on them win those survivor shows in general, because they can burn the fat. They can use glycerol to stay alive. So we'll stay the main fuel. Usually the typical human runs out of fat at about 50 days. Someone who's got a little more adipose tissue will stay low for maybe 70 days. But the average is 50 days. So that's for the average human. After 50 days, skeletal muscle and organ breakdown, so proteolysis in all those tissues will go back to becoming the main fuel, and that will kill you. You get heart failure and your liver will break down. So that's fasting 101 there. When does gluconeogenesis run? So fasting, we're talking about fasting, or diabetes mellitus that's not treated. Type 1 diabetes, someone who doesn't know, kid who doesn't know they're type 1 diabetic. this can happen too. It's like they're fasting. We'll explain that in a while. Normally, it only runs when all the dietary glucose is gone and all the glycogen is gone. No, without insulin around, type 1 diabetes, gluconeogenesis is stuck on. That's one of the reasons that diabetics have high blood sugar, especially type 1 diabetics, because you need insulin to turn off gluconeogenesis. Otherwise, it's going to be stuck in the on position, which is not good. That sugar in the blood causes damage to the small vessels like arterioles, causes them to, it causes protein, it causes glycation to occur inside the walls, and it causes stenosis. And so that capillary bed is not getting the proper blood supply. It becomes ischemic because of the stenosis from non-enzymatic glycation. I don't want to get, it's a long lecture already, so I don't want to get too much in a rabbit hole. But that particularly happens in the eyes, so diabetics, poorly treated diabetics can go blind. Happens in their fingers and toes, so they can lose, their fingers and toes can become ischemic. You can lose and have to have amputation of fingers and toes. Affects the kidneys, so you get diabetic nephropathy. It affects some of the sensory nerves, so you can get a diabetic peripheral neuropathy, where those nerves lose myelin and short circuit and cause pain. So yeah, the glycemia, the hyperglycemia is not a good thing. So yeah, so what exactly happens now? How does glucose get stuck on? So it's that Pepsi K enzyme that gets affected. So where's Pepsi K? It's the step, we haven't come to it yet, and we'll hit this again, but oxaloacetate needs to be converted into PEP. And we don't have there's not a glycolysis enzyme that does that, so we have to use a gluconeogenesis enzyme. And that's called phosphoenolpyruvate carboxykinase, or Pepsi K, I like to call it. Pepsi K is normally turned off in the fed state because you have insulin. And insulin puts the brake on Pepsi K and doesn't let it run. If Pepsi K is running, it'll take oxaloacetate and it'll convert it to phosphoenolpyruvate. It'll drive glycolysis forward. Even if you're eating a hamburger and you've been eating, it'll still drive glycolysis if it isn't inhibited. And most people make insulin and it inhibits it. And specifically, the insulin inhibits something called FOXO1. FOXO1. That's not a zero, that's FOXO1. And that guy is like a brake, and it inhibits Pepsi K from driving gluconeogenesis. So without insulin around, and right now there's no insulin around because our human is starving, and so the brake is removed, FOXO-K, and FOXO-K can drive. In fact, that's the strongest stimulator of Pepsi K is FOXO1. Glucagon, the cortisol, and the catecholamines can also drive this enzyme a little bit, but no, we'll talk a little bit more about it in a bit. But that's it. Without insulin around, the insulin brake on FOXO1 is removed. Insulin is the one who has its foot on FOXO1 and says, you just don't work right now. It keeps it from working. When insulin goes away, it wakes up and it works. And what it does is it goes into the nucleus. It's a transcription factor, and it makes that gene transcribe a whole bunch of Pepsi K, more than normal, and that's going to drive gluconeogenesis very strongly. All right. In general, what does it drive? So some of the key regulatory enzymes are driven by, what drives this thing? Well, we just talked about this already, but glucagon, of course, and its helpers. It's got some helpers. It's got epinephrine, cortisol, and even growth hormone I didn't list are the helpers, but these are the two big ones. And the absence of insulin, as we just said, takes the foot off FOXO1. So what drives glucogenesis in general It's driven by the presence of glucagon or the helpers, and it's driven by the absence of insulin, which is a very important step because it takes the brake off FOXO1. Many of its steps are the reverse of glycolysis, which is probably the best pathway you know. Not all of them, though, because we've already talked about one that's not a glycolytic step. So let's start the story now of gluconeogenesis by starting with pyruvate. So here we go, pyruvate. And this is that same drawing. I'm just taking sections out of it. I made it really huge in Photoshop so I can pull it in little pieces out and they still look really clear. So here's pyruvate. So the first order business, pyruvate is made not in the mitochondria, it's made in the cytosol. So pyruvate has to be gotten into the mitochondria. And there's a transporter called mitochondrial pyruvate carrier that brings pyruvate in. It's not regulated, to my understanding. So great, now we have pyruvate in. So that's the first step in gluconeogenesis. Mitochondrial pyruvate, which way does it go? Now the question is, once pyruvate is inside the mitochondria, it can go two directions. It's used to going this way. There's an enzyme called pyruvate dehydrogenase. It's part of pyruvate dehydrogenase complex. And this enzyme usually grabs pyruvate and morphs it into acetyl-CoA. And acetyl-CoA will combine with oxaloacetate, and then you got citrate, right? That's basic Krebs cycle during the fed state. But we're not in the fed state. So what does pyruvate do? It doesn't go this way. It goes this way. It's converted to oxaloacetate by a new enzyme. You heard it because you've all had biochemistry. But that's PC. That's pyruvate carboxylase. Cool. But how does it know? I mean, I didn't tell you. How does it know? It knows because this molecule, acetyl-CoA, tells it what to do. So, one important fact here. During starvation, the gluconeogenesis doesn't just turn on at 24 hours and it's running full blast. It's slowly ramping up. It starts working at 4 hours, and then by the time it gets to 24 hours after a meal with no other food given, it'll be running, maybe not full blast, but it'll be running very strongly. The process of lipolysis is also following the process of gluconeogenesis together. They go hand in hand, and that's really important because where does this acetyl-CoA come from? What's it even doing here? Because I got an X right there. It's coming from beta oxidation of fatty acids. So, got ahead of my slides. Let's go back and slow down. So, glucogenesis and lipolysis, they both start ramping up about 4 hours after a meal. But by 24 hours, glycogenolysis is done and lipolysis is running very strongly, as is gluconeogenesis. Great. So we're creating, by 24 hours, there's a lot of glycerol and fatty acids in the bloodstream. You could say there's a lot of fat breakdown products floating around in the bloodstream. Great. Fatty acids, what happens to them? They go in, there's 3 fatty acids, they go in, we won't talk about, there's a big old story of how they get through the membranes. We'll cut all that out. But they get in to the matrix of the mitochondria. And that is where something called beta oxidation of the fatty acids is run. So beta oxidation, the end product of taking a carbon off that fatty acid, leads to the final molecule, an acetyl-CoA molecule. And you can make, what is it, like on average, from one carbon you can make 8 acetyl-CoA enzymes. Don't quote me on that. But you also make a lot of these power molecules, NADH and FADH. So those can actually go into the electron transport chain and drive it to make ATP. That's where the energy comes from to drive this gluconeogenesis process. But that's a side note. The key is that lipolysis is ramping up with gluconeogenesis. Lipolysis is making fatty acids and glycerol. So the fatty acids go into the mitochondria and are converted to acetyl-CoA, and we got tons of acetyl-CoA. This acetyl-CoA cannot go through the Krebs cycle, which is, I mean, we're in the fasting state anyway. It's not, nothing's driving it anyway. So what does all this acetyl-CoA do? This acetyl-CoA is an allosteric inhibitor. There is a binding site on PDH, pyruvate dehydrogenase, and acetyl-CoA will bind to PDH and inactivate it. So then PDH can't, if it wanted to, it can't take pyruvate and make acetyl-CoA and then go through the citric acid cycle. Okay? This acetyl-CoA can't go through the Krebs cycle anyway because it's missed this step of going from pyruvate to acetyl-CoA. There's extra carbons involved in that. So we won't get into that. But it's blocked. Why is it blocked? Because we got tons of acetyl-CoA coming from beta oxidation of fatty acids. And if that's not enough, the acetyl-CoA has a binding site on our friend pyruvate carboxylase. It binds to that and stimulates it to convert pyruvate into oxaloacetate. So acetyl-CoA stimulates PC and it inhibits PDH. And that's what makes pyruvate go in this direction. And that's why the Krebs cycle is broken. The Krebs cycle can't run if you don't convert pyruvate into acetyl-CoA. If you get acetyl-CoA from fatty acids, it doesn't count. It can't run through the Krebs cycle, which is off anyway because we're in starvation. Pretty soon this acetyl-CoA is building up like crazy. What are we going to do with the extra acetyl-CoA? That goes into the process of ketogenesis to form keto bodies. And ketone bodies will act as a substitute glucose fuel and fuel everything. Even the brain can use ketone bodies. Red blood cells still can't use ketone bodies, but that's another lecture. I'm sure they got into that in biochemistry. But that's how that little important part works there. So everything I said right here, yeah, beta oxidation makes tons of acetyl-CoA, makes tons of these electron carriers, NADH and FADH2, and we can use those to make ATP by running it through oxidative phosphorylation. Remember, oxidative phosphorylation is a combination of the electron transport chain and the ATP synthase process. Remember, that's a two process thing. All right, great. So, yep, everything we said. Turns out acetyl-CoA can allosterically regulate enzymes that start the normal Krebs cycle entry. The enzyme is called pyruvate dehydrogenase. Acetyl-CoA binds to pyruvate dehydrogenase and inhibits its action. That's what we talked about right there. Great. Pyruvate cannot be transformed into acetyl-CoA, therefore, and it can't go that way. It's starting to build up. Where's it going to go? It's got to go this way. And then there's an enzyme called PC, pyruvate carboxylase, that drives it. It takes pyruvate and converts it into oxaloacetate. Notice one-way arrow. Another one-way. These are all true one-way arrows. So it can't go back. Oxaloacetate can't go back into pyruvate. Are we good with that? Right. Now we have another problem. Oxaloacetate created from pyruvate cannot get out of the mitochondrial membrane. And we need to get it out here into the cytosol, but it's trapped here. So what do we do now? Really simple. We convert it into the intermediate that comes right before oxaloacetate. Double arrow, you can go both ways with this reaction. So it's going to cost an NADH molecule to run it this way, but oxaloacetate can morph into malate. Malate dehydrogenase is the enzyme. And there is a malate aspartate shuttle where a malate can just be shuttled right on out out of the mitochondrial membrane here, and there it is. Great. And malate, there's a cytosolic malate dehydrogenase and a mitochondrial malate dehydrogenase just floating around. So malate will be grabbed by malate dehydrogenase and morphed into oxaloacetate. So pretty simple there. Everything I said right there, malate's moved out, converted back into oxaloacetate. Great. Time to enter some familiar territory because, hey, oxaloacetate, the next step is to convert it into PEP. And that's that's like the last, involved in the last step of glycolysis. We know PEP, so we're almost there. But we need a new enzyme to do it. And this enzyme, for physiologists in particular, I'm more of a physiologist because I teach a lot of physiology. For physiology, this is the rate-limiting step. It's probably the most important regulatory step in the entire gluconeogenesis pathway. And let's look at the enzyme that morphs oxaloacetate into PEP. So, arguably, it's the most important step in gluconeogenesis. Oxaloacetate is morphed or converted into PEP by a powerful regulatory enzyme called PepsiK, phosphoenolpyruvate, or PEP carboxykinase. PepsiK. The boards are using abbreviations now. Let's hope they're biochemistry, they're using abbreviations. For me, you got to know both of these because the boards may or may not. They used to not use abbreviations, and now, for the first time, I saw some abbreviations getting through. You got to know it, though. Cool. So now the question is, what regulates PepsiK? And it is strongly stimulated when insulin levels drop. So the number one regulator of PepsiK is not glucagon, because glucagon stimulates it a little bit as well. And cortisol and epinephrine, the catecholamines, they help drive the reaction this way. This is gluconeogenesis. They help. They're not the big one. The biggest stimulator is the brake on FOXO1. So a decrease in insulin will remove the insulin brake from FOXO1. And FOXO1 will really supercharge this. In fact, FOXO1 is a transcription factor. It floats once it's out from under the foot of insulin and loose. It goes into the nucleus. It goes into the genes, and it stimulates the transcription of the PepsiK gene. And so it's not an allosteric regulator. FOXO1 is not an allosteric regulator. It doesn't bind to PepsiK, but it goes into the nucleus and it makes like an army of PepsiK molecules. And therefore, every little bit of oxaloacetate is morphed into PEP. Normally, without this, you'd have, let's say, 10 oxaloacetates out here, one PepsiK, and one at a time they'd have to get turned into PEP. But now you got like 100 PepsiKs, so they'll all get morphed in, and that's how it speeds things along. So it strongly drives a gluconeogenesis. The lack of insulin or the decrease in insulin strongly drives this enzyme by removing the restraints on FOXO1. Good. Note, contradictingly, biochemistry texts don't say that this is the rate-limiting step. They say the FBPase1 step is. So that's always a problem, and I'm sure the board, I didn't go to the board's writing class in Colorado for biochemistry, but I'm sure they're all well aware of this and stay away from that, or I don't know how they're going to handle that in biochemistry, but that is definitely a problem. Okay, cool. So PEP now, here's PEP down here, phosphoenolpyruvate. It just runs through all those glycolytic steps. I didn't even list them because they're not important to me. They're not controlled, they're not regulated. So I'm just after, I know the boards love rate limiting steps. They love how these cycles are regulated. And so that's what I'm after here. So it goes through all these steps until it reaches, well, we'll do this last two steps. Remember, ultimately it's converted to glyceraldehyde 3-phosphate. That's 3 carbons. So some of the glycerol 3-phosphate is converted into our friend dihydroxyacetone phosphate, which we have some of that coming from glycerol as well. So it's important that both of these 3 carbon molecules come together and form fructose 1,6-bisphosphate. That is a 6 carbon molecule like glucose. So up until now we've had just a half of a glucose molecule, but now we have a early form of glucose, fructose bis 6-phosphate, meaning it's a glucose molecule, but it's got phosphates, 2 phosphates on it. It's not exactly, it's in the fructose form. So, but now we have a problem because we don't have a 2-way enzyme in glycolysis to go from fructose 1,6-bisphosphate into fructose 6-phosphate. Right? We know, remember from biochemistry, there's a dreaded enzyme called PFK1, phosphofructokinase 1, that drives glycolysis. In fact, this is the star of the show in glycolysis, PFK1. So that's a 1-way enzyme. So we need a new enzyme, and we have one. And there's no cute name for this one. This one is called fructose 1,6-bisphosphatase. It's a taser. When you have a taser enzyme, it breaks off phosphates, always. Kinases put phosphates on molecules. Tasers, phosphatases, knock phosphates out. There's a common AKA you need to know for this one, and that is FBPase1. Boards could do either one of these. My test could do either one of these, so you need to know both of these. So we need to get through this enzyme, so we need to know what drives it, what activates this enzyme. And it's interesting because insulin and glucose, or insulin and glucagon, do not bind to FBPase1. Nothing, no epinephrine, no cortisol, no growth hormone, nothing binds to it. So what drives it forward then? It's a decrease in F26-bisphosphate. And you might remember that molecule. That's not an enzyme. That's a allosteric regulating. In fact, that's the most important allosteric regulator of glycolysis. So if that's up high, the step's going to be inhibited. So right now it's going to be, and we're going to go through this giant enzyme here right in a minute. We didn't do that in endocrinology, but I will go through that with you. But right now, let's just get through this. So if you have low F, and that's fructose 2,6-bisphosphate is the name of that, or F26BP. If there's low amounts of that, it'll lift the brake off this enzyme, FBPase1, and FBPase1 is free to do its job, and its job is to take fructose 1,6-bisphosphate and morph it into fructose 6-phosphate. Cool. AMP levels will actually inhibit this. Increased F26BP will inhibit this. These are the inhibitors in this step. Yeah, biochemistry books call this the rate limiting step. So physiology books don't. So on my question, I'd have to say, according to physiology books like Bernie Levy, blank is the rate limiting step. So according to biology books like Leninger, blank is the rate limiting step of gluconeogenesis. And that would be, for that one, it would be FBPase1. Okay, great. So yeah, we talked about that already. Just kind of got ahead of my slides, but from biochemistry standpoint, this is the rate limiting step. From a physiological standpoint, Pepsi K, is the rate limiting step. It's driven forward. Well, what are we talking about? FBPase1, so that's the enzyme again. We're stuck at this step. We're trying to, the whole goal of gluconeogenesis is here. So we're almost there, but we're stuck here. So we need to make sure this enzyme is active. To make it active, we need low levels of F26BP. Okay, so it's driven forward by low levels of F26BP or F26BP or fructose 26 bisphosphate. Not phosphatase. This is a domino. This is an intermediate. It's not an enzyme. And we already talked about that. That'll take the break off of FBPase1 and stimulate it to convert fructose 1,6-phosphate to fructose 6-phosphate. Glucagon inhibits PFK2, and that makes FBP or F26BP, and we'll get to that. We'll get to the real regulation. We'll get to this in a second. Maybe we'll do it right now. FBPase1 enzyme is strongly inhibited by high levels of fructose 26 bisphosphate. Okay, let's finish this up and then we'll come back. So we're almost home now. We've made fructose 6-phosphate so simple. We're going to convert it into glucose 6-phosphate. There's an isomerase enzyme. It's a two-way enzyme. Boring. Who cares about it? So great. Glucose 6-phosphate. And now we're stuck again because this is a one-way arrow. We know hexokinase converts glucose to glucose 6-phosphate, but what converts glucose 6-phosphate back to glucose? Well, we already know from glycogenesis it's the same enzyme, only in hepatocytes and proximal convoluted tubules of the kidney. And that's glucose 6-phosphatase. It's the taser. Where's all the regulation here? Did I forget it? It's not regulated. This enzyme is regulated by a buildup of glucose 6-phosphate, and that just kind of encourages it to push it that way. But insulin and glucagon don't regulate that enzyme. So glucose 6-phosphatase, the taser, what does it do? It knocks the phosphate off there. That was on the number 6 carbon. But it has no allosteric or no hormone regulators. It's simply driven by substrate, by glucose 6-phosphate buildup. Great. Glucose 6-phosphate is then turned into glucose, and glucose is removed from the hepatocyte by a GLUT2 transporter. It's a two-way transporter, so glucose goes right on out. It gets sucked up into the blood, and then glucose goes to every nook and cranny of the body. So that was gluconeogenesis. There you go. There's your review. Hopefully those enzymes are familiar. Hopefully you didn't skate through that class and not remember this stuff, because you're going to have trouble if you didn't. All right, let's talk about that giant enzyme again. What giant enzyme? What are we talking about? This monster right here. It's a monster, but it's the most important regulator, really, of glycolysis and gluconeogenesis. It's super important. And it's a beast. I call it the giant bifunctional enzyme. What's its name? Its name is FBPase2 PFK2, or fructose bisphosphatase 2, or phosphofructokinase 2. But I'm sure the test, everybody just calls it FBPase2 and PFK2. So what is it? It's a gigantic polypeptide chain, and it's got 2 enzymes inside that chain. So 2 enzymes in this gigantic molecule. Wow. So who cares about it? Well, who cares about it? This is what makes, this beast of an enzyme is what makes fructose 2,6-bisphosphate. F26BP is the most important regulator of glycolysis, and it's an important regulator of gluconeogenesis as well. But it's the most important regulatory molecule of glycolysis. This molecule, which is created by, these are the enzyme parts of the beast. There, it's created right there. It's not a member of gluconeogenesis pathway or glycolysis pathway. It's a regulating molecule. It's not an enzyme. It's just an allosteric regulator. It has the ability to bind to things, to enzymes, as we'll say. Recall that normally it drives PFK1. PFK1 that's phosphofructokinase 1. That is an enzyme in glycolysis, and that is the rate-limiting step of glycolysis. and that's going to convert fructose 6-phosphate into fructose 1,6-bisphosphate, right? If we go back, do I have it? Fructose 6-phosphate, if we're going through glycolysis, we're going this way. So there's PFK1. Normally, it converts fructose 6-phosphate and fructose 1,6-bisphosphate. And it is normally driven by, there is fructose 2,6-bisphosphate. It's normally driven allosterically by that enzyme. I have it blocked with red because we're in gluconeogenesis. And we'll come back to that. All right, great. So yeah, what else? So the $64,000 question is, where does F26BP come from? This most important regulator of glycolysis, an important regulator of gluconeogenesis. Where does it come from? It comes from that giant enzyme, right? It comes from the giant bifunctional enzyme, which lives in the hepatocyte cytosol, floating around, looking for trouble. And it is a giant polypeptide chain, and it contains in the same chain two enzymes, PFK2, not PFK1, and FBPase2, not FBPase1. PFK1 and FBPase1 are part of the gluconeogenesis and glycolysis pathways. Okay, so has two parts. We said that. It's got a PFK2. So this PFK2 turns on or stimulates or drives glycolysis, a good study slide right here. It inhibits gluconeogenesis. How does it drive glycolysis? It makes F26BP. Fructose 2,6-bisphosphate is an allosteric regulator of fructose 6-phosphate. Or no, it's made from, where does it come from? It's made from fructose 6-phosphate. What does it do? It drives PFK1, which drives glycolysis. Let's look at it. So here's glycolysis. There's glucose 6-phosphate, fructose 6-phosphate is here, fructose 1,6-bisphosphate, etc., pyruvate. So some of the fructose 6-phosphate can come out of this pathway. If PFK2 is activated, it can be grabbed by this giant enzyme, the PFK2 part, and converted into our super important regulator, F26BP, or fructose 2,6-bisphosphate, the master regulator. That's where it comes from. So yeah, in glycolysis. One enzyme turns off glycolysis. So the little lines mean the enzyme is active. So the FBPase2, if that one is activated by being phosphorylated, but if it's activated, that will stimulate gluconeogenesis. How come? Because it takes the break off of gluconeogenesis that is put on it by fructose 26-bisphosphate. F26BP, remember, that's a brake on glycolysis. And so if FBPase2 is on, it inhibits the creation of fructose 2,6-bisphosphate. It blocks the step right here. We could have put the arrows here, the little lines to make this active. We could look at either way. But yeah, it blocks it right there. And therefore, yeah, that's all I need to say about that. So it stimulates gluconeogenesis, converts F26BP back to where it came from, fructose 6-phosphate. So that's what it does. So what is FBPase2? It blocks this way, so you don't have any more, you're not making any more fructose 2,6-bisphosphate. But the fructose 2,6-bisphosphate that is around is grabbed by FBPase2, and it's morphed back into fructose 6-phosphate. In fact, that helps drive gluconeogenesis, because this is the direction gluconeogenesis goes. See how that works? And what controls it more specifically? Because I've just been saying, well, insulin will turn on PFK2, and glucagon will turn on FBPase2, but there's a little more to it. So it's controlled by phosphorylation and dephosphorylation. So, yeah, insulin, what does it do? Let's look at it. Insulin is ripping off a phosphate right here. And it has a little henchman that does the work for it, phosphoprotein phosphatase 1. I'll be happy if you know insulin will rip a phosphate off this giant molecule. So it dephosphorylates the giant PFK2B FBPase2. Great. Glucagon does the opposite. Glucagon's over here. That works through a cyclic AMP-dependent protein kinase. You don't need to know that. But you do need to know that glucagon will steal a phosphate from ATP, and it sticks it right there. So, and this is the same enzyme, right? It's just two different states here. That's what this picture is showing. There's not 4 enzymes in this. There's only 2. It's just showing 2 different states with this drawing. So, bottom line, glucose phosphorylates the giant. If the giant is phosphorylated, who's active? If there's a phosphate on here, FBPase2 is activated. That's going to block the formation of fructose 2,6-phosphate, and go a step further, it's going to take fructose 2,6-phosphate and put it back where it came from, which is fructose 6-phosphate. See how that works. If a phosphate is ripped off and replaced by a hydroxyl group, that's what insulin does. Look at the phosphate is out, it's ripped off. Phosphophosphatase 1 does that. If PFK1 is or PFK2 is active and FBPase2 is inactive, this one rules. What does PFK2 do? That's going to grab some of the fructose 6-phosphate and morph it into the super important regulatory molecule, fructose 2,6-bisphosphate, or F26BP, the master regulator. Who makes that? You should know that. PFK2 does. How does PFK2 do it? What turns on PFK2 to make F26BP? Well, it has to have a phosphate ripped off. Who rips off the phosphate? Insulin or glucagon? Insulin rips off the phosphate off this molecule, slaps a hydroxyl group on, right? And then what does fructose 2,6-bisphosphate do? It's an allosteric regulator. It stimulates PFK1. That's a glycolysis enzyme, right? It's rate-limiting step. Turns fructose 6-phosphate into fructose 1,6-bisphosphate. In other words, it drives glycolysis. What else does the same enzyme do? It inhibits our gluconeogenesis enzyme, FBPase1. So going from fructose 1,6-bisphosphate into fructose 6-phosphate is blocked. See how that works? In this class, we're only talking about this one being active. We're not talking about PFK2 being active, because we have glucagon around, and glucagon is going to stick a phosphate on there, so it's going to activate this one. And you could activate that one and deactivate that one, but this is the way the author drew this thing. And again, with FBP2A active, what happens? You don't make any more fructose 26-bisphosphate. In fact, you take it and you put it back where it came from, fructose 6-phosphate. See how that works? That is the giant enzyme explained the best I can explain it. And this is everything I said already, so I don't think I need to talk about it again. I'll go slow if you want to read through the slides. But yeah, everything I said. Okay, I mean, this is where we are, I guess. Glucagon phosphorylation of the giant, we can hit that one more time because that's what we're talking about. And why are we doing this? Because we're trying to get past that step. We need to have our FBPase1 activated, so we can't have fructose 2,6-bisphosphate around because that messes things up. If it's around, it inhibits our FBPase1. We don't want that. So we have to have glucagon around to fix that problem. So during fasting, glucagon rules. Glucagon will enter the hepatocyte, trigger phosphorylation of the giant. Right here, it sticks a phosphate on the giant, which wakes up the FBPase2. That is going to, this also deactivates PFK2. So you can't have both of these enzymes on at the same time. Great. What happens next? Everything I've already said. It grabs F26BP, converts it back to fructose 6-phosphate. So now fructose 6-phosphate is back in the gluconeogenesis pathway. Cool. Indirectly, what does FBPase2 do? So that's our friend here. Well, indirectly, without fructose 2,6-bisphosphate around, that allosteric inhibition of FBPase1 will be over. And so FBPase1 is no longer inhibited like it is up here. It's released, and we can drive gluconeogenesis once again. Junk pile. So we're not going past here. I think this, what is this, like 80 slides or 90 slides? This is like the 10th version of it. So I think I got a pretty good story down pretty good for a physiology teacher, right? Not a biochemistry teacher. I think I hopefully you can understand it. Biochemistry is just tough. But it's definitely on boards, and it's definitely mixed in physiology, and it's definitely on your final in board review class. So send me emails if you have questions. Listen to the recording back again. And good luck. See you later.